really happy to introduce Pauline Yu to you. Pauline is nearly new, or the other term we like to use sometimes is gently used faculty here at Evergreen. She's been here, this is her second year. She got her PhD in biological sciences at the University of Southern California in the Division of Marine Environmental Biology. She did a postdoc at University of California, Santa Barbara, which involved 3 seasons in Antarctica. And I've been assured that those were the summer seasons, not the winter seasons. And she's also done a lot of thinking and work around ocean acidification. And in particular, you might want to check out a really lovely article by Devon Barrowman in the Cooper Point Journal from a year ago, January 2015, on ocean acidification, and in an interview with Pauline and also with Eric Tucson and several other greeners about this issue and the things that people are doing at Evergreen about it. And so she's here with us to talk about ocean change, the carbonate trajectory of ocean climate. Thank you, Pauline.
Thank you, Ruth, for inviting me. Before I get started, I also want to point out that, unfortunately, due to scheduling conflicts, almost concurrently, there is a coming together session happening in the library right now. And I know this is an issue that's important to a lot of people in this audience. And unfortunately, you're here with me and not there. But at the same time, I encourage you all to sort of follow up on what you're potentially missing there, because I think it's really important to think about the climate and trajectory of Evergreen as a learning community and how we face issues of inequality and power differentials, and especially as it relates to race. Okay, so let's get started. So my talk today is going to be integrating a lot of the biological and chemical consequences of carbon dioxide as it's been introduced into the atmosphere in general on the organisms that live in the oceans. So if we think back to the great lecture by Abir at the very beginning of the quarter, and think about how through Earth's geological history, there have been periods where there have been higher levels of carbon dioxide than there are now, but those changes happen very gradually. And so organisms were able to adapt on evolutionary timescales. And then we can also think back to Gerardo's talk several weeks ago about how ocean and atmosphere and climate and carbon dioxide all are deeply, intricately interlinked. And so I'm going to move from that really planetary, global scale to much more small scales, thinking about how organisms in the ocean are going to be affected by those big global planetary scale changes. So here's my outline. I'm going to review some really crucial concepts that relate to chemistry and biology. And then I'm going to move from the chemical bases of these changes occurring to the biological consequences. So to start out with, I want to review the concept of pH. I know many people are familiar with it, but it's not something that many of us think about on a daily basis. And when we think about acid-base relationships, it's related to this highly quantifiable property. And so we think about water. Water has 2 hydrogens and an oxygen. And that covalent bond between hydrogen and oxygen is somewhat fluid in that once in a while you can have one hydrogen pop off and add itself to another water molecule. And so both of these states, creating that free hydrogen proton ion or creating what's called the hydronium ion, the extra H, H3O plus in water, this happens normally, all the time, back and forth in an equilibrium. So pure water is defined as neutral. Because we are water-based organisms, we are the water planet, we define everything, a lot of things, against water. So in this case, we define pure water as neutral. The concentration of these hydronium ions as 10 to the minus 7 moles. We set that as the middle. And then from there, everything is either higher concentration than that or lower concentration than that of hydronium ions in water. So when we're describing that unit we know as pH, we're using a negative logarithmic scale of this concentration. So if it's 10 to the minus 7 moles of these hydrogen ions, then as we go down the pH scale, going from 7 to 6, we're actually increasing that concentration of ions. It's going from 10 to the minus 7 to 10 to the minus 6. As the numbers increase on that pH scale, it's actually going in the other direction. So as we go from 7 downward to 0, things are becoming more and more acidic. There's a higher and higher concentration of hydronium ion. And as we go higher from 7 to 14, we're decreasing the concentration of hydronium ions in aqueous solution. So keep in mind, log scale means that there's a 10-fold difference between something at pH 6 and pH 7. We also refer to that as an order of magnitude. So in terms of how pH affects our physiology, because most of our cells are based in water, that they actually are spending a lot of energy trying to regulate how much hydronium ion stays inside or outside the cell, depending on the particular needs of that cell. It's not perfect. It's not kept at precisely pH 7, just like pure water. We're actually slightly basic. So the pH in human blood and inside cells is approximately 7.4. On this scale, you can see here that seawater is even slightly more basic. It's at pH 8.1. In the spaces between the cells, we'll call it extracellularly, there's actually a wide range in biological organisms. So in the space of our stomach, which is not inside of our cells, we can maintain this very, very acidic, very, very low pH, pH 2 environment. But that's existing outside the body of each of your individual cells. And then there are examples of animals where they can maintain or survive in very high extracellular pH environments where it's very basic outside of the cells in their bodily fluids. So there's a very intricate regulation that goes on between keeping that intracellular pH at this very narrow, confined concentration of hydronium ion and the extracellular matrix. So that always gets tightly controlled between the inside and the outside of the cell. And this is done by active transport. So the cell membrane is what we call a semi-permeable barrier. So permeable means things can go right through. Semi-permeable means that only certain things can go through. So hydronium ion, anything that's ionized in general, like salts, they can't pass directly through the membrane. The membrane blocks that passage. But there are special proteins embedded in that membrane that act as channels or pores. So they selectively allow things in and out. And so if you have an acidic environment outside the cell and a more neutral environment inside the cell, each cell is actively maintaining that differential inside and outside that cell. And in some cases, they can passively allow a little bit of acid into the cell if the cell needs it, or they can actively use energy and push those extra hydronium ions out of the cell into the extracellular space to maintain the appropriate pH inside the cell. Now, maintaining this pH is really important. The entire body of most living organisms need to maintain a certain balance of ions in cells so that everything can function properly. So for us terrestrial organisms, the only way that fluids enter our body is largely through the mouth. You can breathe in water vapor, but largely through the mouth. Whereas marine and freshwater organisms, they're completely surrounded in this aquatic medium, and so they're constantly trying to regulate the ratio of these salts and hydronium ions passing in and out of their bodies. In most cases, the marine organisms, since that external environment of seawater is slightly more alkaline or basic than inside of their cells at this slightly lower pH, they want to keep all these hydronium ions inside their cells and prevent them from leaking out into that slightly more basic environment. In a lot of organisms, you have gills and mucous membranes where this regulation is occurring in and out of cells and in and out of the body. However, this is also energetically expensive. So there are some marine organisms, in order to save energy, they allow their cells to actually conform, in this case, osmoconform between the external seawater medium and the inside of their cells. And in that case, that usually applies to marine invertebrates because their skin tends to be more permeable than that of, say, fish, and definitely those of marine mammals such as whales and seals. So if we think about the scale of possible changes in the pH of the ocean, we have to think about minute changes on the pH scale that are actually happening at very much larger changes relative to percentages and proportions. Because remember, going from pH 8.2 to 7.2 is a tenfold difference. So slight changes in the pH measurement, 1 unit, actually represent these much larger changes in concentration overall. So if we compare between certain higher pH regions of the ocean versus slightly lower pH areas of the ocean, or looking at global averages over multiple decades, we can report it as, okay, the average pH of the ocean has gone from 8.2 to 8.1. Well, there are a lot of climate deniers out there who are going to say, well, that's a really tiny number. I mean, I don't see what the problem there is. But if we actually calculate the concentration change in these ions, you can see, you know, a third-fold change or a two-fold change. So going from pH 8.2 to pH 7.9 represents a doubling of the concentration of ions. That's biologically significant. This is not a small, it seems like a small number change, but it's actually a very biologically significant change because the insides of cells want to be within a narrow range of pH in order to operate properly. So, you know, why is this important? Well, we can think about how it affects the biochemistry and function of cells. So, for those of you who haven't had much of a chemistry background, a lot of proteins are highly dependent on interactions with surrounding water molecules inside of the cells, and they depend upon this loose energetic bond called the hydrogen bond. So it's one of the things that makes water cohesive is this loose, low energy interaction. And proteins rely on this low energy interaction to be able to make slight changes in shape to function or interact with other proteins or be able to actually fold in on themselves from a linear protein molecule into a particular 3D shape that gives it function. So within the cell, all of these functions are tied to and regulated by things like the interactions with water around it, and hence all the things dissolved in that water, like salts and the pH. Another thing that's very important for organisms is the process of digestion and regulating the pH that occurs during digestion. So if we look at a single-celled organism, they have what we can call a temporary stomach. They can take in a little bit of food into an intracellular pocket, and then inside this pocket, they can make it temporarily acidic in there to digest the food, and then with the use of digestive enzymes that operate at these low pHs, they can break down the food into its individual molecular components. The cell can utilize those components like sugars and amino acids and fatty acids. And then once it is done digesting the usable parts, it can then eject the leftover components out into the rest of the environment. But before it does that, it's going to change that pH from low pH back to sort of normal pH. And that's what happens in our stomachs as well. We take in relatively neutral pH foods, we acidify them in our stomachs, and then in the intestines, they gradually come back to a more neutral pH. Furthermore, cells are extremely reliant on maintaining particular osmotic gradients in these intracellular components known as mitochondria. Mitochondria are what is known as the energy factory or the fuel cell of all eukaryotic cells, so cells with nucleuses that contain the genome, and also there are processes in bacteria and cells without nuclei where they rely on these pH gradients to help them generate energy. So in particular, the mitochondria, the energy factory, is using the digestion of sugar and the maintenance of pH gradients across a barrier to harness the energy of protons moving from an acidic space into a less acidic space, and harnessing that chemical energy to turn it into chemical energy that can be used elsewhere in the cell. So now that you have sort of an overview of the biological effects of pH, I want to do a little aside to address a question that was actually brought up during Gerardo's lecture a few weeks ago. And somebody asked about acid rain and how that may or may not relate to ocean acidification. So acid rain has its most pronounced effects on terrestrial ecosystems because the terrestrial landscape holds smaller amounts of water than the ocean. As you recall from Gerardo's lecture, most of the Earth is seawater. And if you're adding the small amount of water from rain, acid or no, into that vast pool of ocean water, it's going to get diluted out. The ocean has buffering capacity to neutralize any acidity in the rain. The land does not have that same buffering capacity. So acid rain is caused by gaseous emissions from industrial activities. It's produced when we release sulfur dioxide and nitrogen oxide gases into the air. They're reacting with water vapor in the atmosphere, and then you have these clouds where they're effectively full of tiny amounts of sulfuric acid and nitric acid that then rains back down on the Earth. So in this map, it shows different regions of the country where there is more acidic rainfall. We're lucky on this side of the country that the rainfall is relatively natural at its natural pH, which is slightly acidic. It's slightly acidic because of interactions with carbon dioxide in the atmosphere, which I'm going to go into more detail about shortly. But acid rain is interacting with the carbon dioxide, the sulfur dioxide, and the nitrogen dioxides, so creating this mix of different acids that then rains on the Earth into the rivers and streams and lakes and onto soil, where there's less buffering capacity because water on land is mostly coming from precipitation or snow. All of these come from the atmosphere. If it's acidified, it stays in that environment, and then it tends to poison the organisms there. So very different phenomena. And fortunately, because we have put in place regulations to change the amount of emissions, we actually have seen over the past few decades a decrease in the amount and severity of this acid rain. It is still occurring, but it is a lot less severe than it was in the 70s and 80s. So going back to phenomena in the ocean, we're going to switch gears and get into some more of the chemistry. So in this data from the Intergovernmental Panel on Climate Change, we see a lot of data from time series measurements. So time series measurements are where people go out into the ocean at particular spots, and throughout the year, they're always measuring the same parameters over and over again and looking for long-term trends. So in this top figure, this black line actually represents the concentration of carbon dioxide in the atmosphere as it's slowly rising. And we did discuss in Gerardo's lecture a little bit why that's always a zigzag. It's not just a flat line, because of the activity of plants on a seasonal basis, taking up carbon dioxide, and then during the winter, they're not photosynthesizing as much and they're not taking up as much. So that level goes up and down. The green, yellow, and blue lines represent measurements being taken in the ocean at different remote open ocean research facilities. So the green line is data from Bermuda, the yellow line is data from Hawaii, and the blue line is data from the Canary Islands. So far, far away from any coastal pollution, people are trying to look at what may be the, a representative measurement of what the ocean average carbon dioxide is. And so in this first graph, we're looking at measuring the partial pressure of dissolved carbon dioxide gas as it comes from the atmosphere and mixes with the seawater at the surface, and looking at that fluctuation. So it also fluctuates in the ocean just like it does in the atmosphere, although the magnitude of that fluctuation is actually quite a bit higher, and I'll go into a bit more detail about that later. The other thing is that they're taking multiple measurements of the ocean. So they're also measuring the pH of the ocean, and then they're measuring another parameter of the ocean of dissolved carbonate ion. And all three of these measurements move together in sync with the oscillation of carbon dioxide in the atmosphere as well. We just see differences in magnitude. But all these trends give the same result, that the dissolved carbon dioxide trend in the ocean is that the average is increasing despite the up and down every year, that the average pH is decreasing over the past few decades, and that the concentration of carbonate ion in the ocean is also decreasing. So at the same time, the oceans on average are warming. So we're looking here at global averages, but at the surface level, and trying to get a sense of, okay, what is the surface pH level in certain regions of the ocean, and what is the surface temperature at certain regions of the ocean. And so over here, we can see that over the globe, there is a wide variation of temperatures. The average temperatures tend to be much warmer at the equator and much colder at the poles, unsurprisingly. But similarly, we have a variation over the surface of the globe in the surface pH. And so we can see that the scale goes here from what's actually present on the surface of the Earth now is from just under 8.4 all the way down to just under 8.1 in terms of range of pH. In terms of temperature, we have this range of approximately 20 degrees Celsius. The projected change for the next 100 years is an increase in temperature of 2 degrees Celsius. The projected change of pH is encompassing what is actually on Earth, almost all of the differences on Earth right now. That projected change in pH is going to be approximately 0.3 units of pH over the next 100 years. So we talked about how it's how many fold change, so it's going to be more than 30% fold change, it may be 100% fold change, it will be at least a doubling of the concentration of hydronium ion on average over the entire globe across all these different regions. So we know a lot about how increasing temperature affects organisms. We don't know nearly as much about how average decreasing pH is going to affect organisms. So now I'm going to get a little into the nitty-gritty of the chemistry. So I have little cartoons of molecules here. They're not to scale. So we have carbon dioxide molecules from the air that are entering the water through either just mixing or through the activities of organisms right near the surface. These are our little water molecules. So we have this population of molecules. It's a mix of water and carbon dioxide. So if we just, for the sake of math, if we look at 3 water molecules and for every carbon dioxide molecule, they can undergo this reaction where one of them can give up its, one of the water molecules will give up its hydrogen to another water molecule. Another one will give up its hydrogen to a carbon dioxide molecule. And so through this equation, we end up with what we call a bicarbonate molecule. So it's going from a dissolved gas into being an ion dissolved in water. It carries a negative charge with it. And because in chemistry we like to keep our charges balanced, here everything is neutral. Here we have 1 hydronium, 1 acid unit for the base unit, the bicarb. So this is the bicarbonate, just like in baking soda. If we react that water further, we can end up with the addition of the hydrogen here onto this second water molecule, and we can end up with 2 acid units of water molecule with 1 ion of carbonate. So this now carries a charge of -2 that balances out the 2 plus protons on this side. So there's all this sort of back and forth trading of hydrogen atoms that goes on in water. So this water reaction is constantly going on. It constantly goes back and forth. So this reaction can go backwards and forwards. It can convert back to this. It can convert back to this. All of this is happening in dynamic flux. So when we go in as chemical oceanographers and make measurements of what the ocean chemistry is, when we go in and measure pH, we are directly measuring the presence of and concentration of these ions. Another way that we can make this measurement is we can directly measure just the carbon dioxide, just that neutral carbon dioxide molecule dissolved in water. So remember, gases can be in water without reacting, so some of that carbon dioxide remains unreacted in water. We also have 2 additional measurements that we take to assess how much carbon dioxide is in the water, and that is measuring the total dissolved carbon, which is what it sounds like, measuring this dissolved carbonate charged ion, and we measure the total alkalinity, which is trying to look at, as opposed to the pH measuring the acid component, the total alkalinity is trying to characterize the total base, the negative charge component that's in the water. So if we go back to that figure I showed from the IPCC, we're actually simultaneously measuring 3 different components of this system, of these reactions that are happening. And while these measurements are interrelated with each other, there is an algebraic equation that describes the behavior of carbon dioxide in seawater with 4 different variables, the ones I just showed you. Each of these can be independently measured. And then, the more you measure, the more robust your calculation is of what the actual state of carbon dioxide in the seawater is. So additionally, carbon dioxide behaves somewhat unusually when it's dissolved in seawater. It tends to be better at dissolving in seawater at lower temperatures. So here we have a graph of temperature along this axis and pressure along this axis. And this line here shows the interaction of carbon dioxide across the state change going from gaseous carbon dioxide to liquid carbon dioxide. Most of you probably have never encountered liquid carbon dioxide. It only occurs under high pressure. So this carbon dioxide gas, as it mixes into the seawater, you can actually mix more of it into seawater at saturation. So this graph is trying to show the maximum concentration of carbon dioxide you can get into seawater under particular conditions. So under higher pressure, under lower temperature, you can introduce more carbon dioxide into water. That's why when you drink your soda water or whatever fizzy beverage you prefer, when you drink it cold, it's got more fizz to it. When it's warmed up and it goes flat, that's because all that carbon dioxide is no longer saturated in it. It's trying to escape out of the liquid. So keeping in mind that carbon dioxide behaves in these strange ways under conditions of low temperature and high pressure, we can think about certain environments where this may affect the organisms more so than at the temperatures that we find comfortable. So in polar regions, we can imagine that at the surface, it's extremely low temperature, but low atmospheric pressure as well. So if the saturation boundary of carbon dioxide is here, it's not completely saturated, but it's closer to saturation than if the temperature is much higher. In the deep ocean, you have both low temperatures and high pressure. That's another environment where carbon dioxide in this gray zone tends to be saturated. So you can have these environments where the carbon dioxide load is as high as it can chemically be. And in terms of sources of carbon dioxide, there's natural sources in addition to the ones coming from the atmosphere. So in the Earth's functioning with geological activity, you have very active tectonic activity at the bottom of the ocean. You have trenches, you have vents. All of these are places where seawater is seeping into the cracks of the earth, mixing under heat and pressure, and then being released back into that seawater above. So there are places where there's geotectonic activity that also can generate carbon dioxide through these inorganic processes and release it into the seawater. What's really important is that respiration, the biological contribution, is what causes those oscillations that we see in the atmosphere and in the ocean. So under conditions of illumination, under photosynthesis, we have organisms that take the energy of light, they take up carbon dioxide in its gaseous form, combine it with water, and they can generate stored energy in the form of sugar, and then they generate oxygen as a waste product. So photosynthesis is this energy producing and storing process used by a lot of organisms on Earth. It's not just plants. there's phytoplankton, there are bacteria, all of these different varieties of organisms can engage in this process of photosynthesis. In the ocean, it's only going to occur during the day, and it's going to only occur in the upper layers of the ocean where sunlight can penetrate. So if you think about how that depth of the ocean constitutes this huge volume on Earth, it's really this skin layer of the ocean where the light can penetrate, where this photosynthesis is occurring in the ocean. On the other hand, respiration, what all of you are doing right now in this room, you are converting carbon substrates such as sugar, and you're taking in oxygen, and you're turning it back into that carbon dioxide and water, and you're using that energy to do other things, run all your cells, move, go through the process of breathing and taking in more oxygen. So this in the ocean is occurring everywhere where oxygen and metabolizable organic carbon are present. So that actually encompasses a lot more of the ocean than where photosynthesis can occur. So by volume, there's a lot more respiration going on than photosynthesis. In this process of photosynthesis, because they're removing carbon dioxide from the external world and internalizing into cells, this conversion of carbon dioxide into sugar tends to make seawater more basic. So sugar is a neutral molecule. It's turning it into sugar and starch, storage, so it's actually removing carbon dioxide and removing it from getting in those interactions with water molecules and making the surrounding seawater more basic. So on the converse, respiration is generating more CO2, and so it's always constantly slightly acidifying the surrounding water. So in the environment, there's always this trade-off between photosynthesis and respiration, and there are certain coastal systems where we actually see these oscillations and fluctuations happening on a daily basis. So for instance, we have data here from a coral reef, which is being measured for pH in real time, and every day there's an upswing in pH, so it's becoming more basic, and that's happening when the corals are photosynthesizing, and then at night, that photosynthesis stops, and only the respiration is going on, so that pH becomes more intense. It gets more acidic at night, so it fluctuates like this on a daily basis. And estuaries, kelp forests, tide pools, all of these are highly productive systems where there's photosynthesis going on, and then the organisms that are consuming that photosynthesis constantly breathing. Additionally, you can have the products of that photosynthesis happening in that upper layer, and all that organic carbon trickles down to depth, as does some of that oxygen, and all the organisms out of the illuminated parts of the ocean are taking up that carbon, eating it, and breathing out more carbon dioxide. So deep water, away from where photosynthesis occurs, tends to have more of an acidic ocean water because it's not being balanced out by the contribution of photosynthesis. And so here is data from a series of cruises conducted by the National Oceanographic and Atmospheric Association, NOAA, the government organization. So they went up and down the West Coast, so we are way up there. They went on cruises out along the coast, and they measured the pH as they went out along the coast. And they found that there are certain areas where even at the surface, some of that deep water has been pushed up due to ocean currents mixing water from deep up to the surface and bringing some of that much more acidic deep water up to the surface. And so these reddish patches along the coast are areas where deep acidic water has been pushed up to the surface. And that's effectively the ocean burping carbon dioxide back out into the atmosphere. And I don't know why this one disappeared. There we go. So a really important thing to think about, given the oscillations I just discussed, that oscillations happening on a daily basis and oscillations happening on a yearly basis, is that you have that oscillation going in a particular pattern, but as the oceans, the trend continues that the oceans become more acidic, that oscillation is going to move directionally. So we're talking about a range of natural variation right now, which is what we call the present day envelope between the highs and the lows. Just like we have the lows of temperature right now in winter, and this past summer we had the highs in the 90s in Washington. So we have that normal range, but as ocean change occurs, that range, that oscillation is going to continue to move in a particular direction. And what we are estimating from these models is that this current oscillation, the lowest that organisms currently see, will actually become the high by about 2035. So that the magnitude of change is not going to change on an annual basis, but the highs and lows are going to get shifted downward as we go. So this present day envelope, this current present day range is going to be outside of what becomes the new normal by 2030. So these are things that we're thinking about in terms of how organisms are going to be able to handle these changes in their environment, because these changes are happening relatively rapidly. This is only 20 years from now. That's the estimate of how quickly this is going to happen. And evolution happens on these much longer time scales. In Abir's talk, we talked about geologic time. We talked about tens of thousands, 100,000s, millions, tens of millions of years over which the natural changes in Earth's climate have happened. And here we're talking on the scale of decades. So I'm going to switch back to biology and talk about carbon dioxide and physiology further. So we have the different components of the carbonate system that I discussed. So CO2 gas, as in air or dissolved in water, is part of respiration. As it undergoes that reaction with water molecules, we form that bicarbonate. Well, that bicarbonate, because it is a charged molecule, because it is dissolved in water, it can take place inside the cells and engage in acid-base homeostasis. So helping the cell to stay within that range of preferred pH, around 7.4. So if the cell needs to be more basic inside, it can take in this dissolved bicarbonate. If it needs to become more acidic, it can push that out into the extracellular space. Another way that carbon dioxide is traveling around in the body is that it gets dissolved into the water, it gets converted to this bicarbonate, and then it can be transported, dissolved in blood, to other parts of the body, where it can be converted back to carbon dioxide gas to be exhaled. So in this diagram with the fish, I have it drinking in the seawater with the water, with the bicarbonate, with the carbon dioxide, and it's excreting out excess carbon dioxide. It's doing this chemical conversion within its body and then also excreting the bicarbonate. The other forms of the bicarbonate and the carbonate ion are heavily involved in the process of biomineralization. And so I'm going to go into more detail about that. But the formation of shell material by living organisms. We suspect that carbonate ion also has a role in acid-base homeostasis. It's one of those things that people are now doing more investigations into because of the phenomenon and concerns over ocean acidification. So calcium carbonate. So this is two charged ions. The carbonate is a -2 ion. Calcium is a +2 ion. And it can exist as a solid mineral and in a dissolved state. So marine organisms use these components to make shells. And these shells are found in two different mineral forms, aragonite and calcite. So here we have examples of a coral and alga. So this is a photosynthetic life form that deposits hard calcium carbonate deposits. And then we have a little abalone here, so it's a snail. So all of these organisms deposit their calcium carbonate mineral as the form aragonite. We also have calcite. This is an image of a particular type of phytoplankton called coccolithophores. They secrete these tiny little plates that are made out of calcite. Sea urchins and sea stars and all other echinoderms deposit their shells as calcite. And then also crustaceans, so this little crab. So all of these biological organisms are secreting calcium carbonate to make shells, but they're doing it in slightly different mineral forms. So how is that chemistry occurring? So we had our dissolved hydronium ion and carbonate ion. Well, there's actually quite a lot of dissolved calcium in the ocean. So all this dissolved calcium in the ocean is free to then combine with these free carbonate ions in the equation as we give it here. So this is the soluble or dissolved form of these two free ions, and then this is the combined solid mineral or precipitated form of the ion, of the ions into mineral. And we describe the chemistry of them going back and forth between dissolved and precipitated with this Greek letter omega. It's just a ratio that we calculate describing the rate at which the solid dissolves or the dissolved stuff precipitates back out into a solid. So with this ratio in mind, we can have different states. We can have an omega value greater than 1, which means that the chemical process tends to go towards precipitation and makes it easy for shells to be formed. On the other hand, we can have it going in the opposite direction. We can go in the direction of dissolution, where omega values are less than 1, so they can be 0.5. And in this case, the chemical tendency is towards dissolving, and that makes it more difficult for organisms to form the solid. And under conditions of low partial pressures of dissolved carbon dioxide, Under conditions of high pH, fewer H protons dissolved in water. Under conditions of warmer temperature, we're more likely to have these omega values greater than 1. And so conversely, as we increase the levels of PCO2, or partial pressure of carbon dioxide, and we lower the pH, we tend, and also under colder temperatures, we tend to have the chemical reaction going towards dissolution. So you can think about global warming generating this paradox, because we'll have warmer temperatures, but more carbon dioxide. So organisms are going to have to face that sort of contrasting, competing dynamic in terms of how they're able to form shells and skeletons. So if we take, for example, this sea urchin larva, so this is planktonic phase of the life cycle of a sea urchin. These are only at most a few millimeters in size. Usually they're quite a bit smaller. And they deposit an internal skeleton made up of this calcium carbonate, which I can visualize here using polarization microscopy. It makes the crystal structure of this calcium carbonate show up and makes this flesh that covers it invisible to light. And if we can imagine if we can imagine all of the reactions that are going on internally in the body of the organism at this time. It's taking up calcium ion and combining it with carbonate. It's taking up all of this bicarbonate and carbonate and carbon dioxide into its intracellular tissues and trying to balance out its own homeostasis, trying to maintain the right balance between the external seawater medium and what its cells need to thrive, and then also trying to deposit and precipitate this mineral all simultaneously in order to go about its life cycle. And if we look at, so we looked earlier at global patterns of pH, we can look at global patterns of calcification and see which regions of the globe it's easier for organisms to calcify. So we've color-coded it. In this case, red means that our omega value is higher. It's much greater than 1, whereas as it gets closer to 1, where it's sort of equally likely to dissolve or precipitate, that the Arctic and Antarctic, the polar regions, are much closer to this value of 1 because of the cold water. Whereas closer to the equator, where these coral reefs are, we tend to see these higher values. These warmer temperatures allow organisms to calcify much more easily. So here we have the range across the Pacific coast along the margin here. So these values are above 1, but they're closer. And then in these tropical regions off of northern parts of South America and Central America, they're actually somewhat more acidic, the organisms are less able to create this biomineral than they are on the other side of the Pacific, closer to the Great Barrier Reef, where we have lots of coral activity because it's easy to calcify in those regions of the ocean where it's nice and warm. So I'm going to switch gears a little and look at specific research studies that have been done on different aspects of the interaction of carbon dioxide with the local environment. And We approach this through a variety of different ways. We can look at the possibilities of how organisms will respond to global change by looking at gradients that are naturally occurring. So we can see that there's a latitudinal gradient from the tropics to the poles in this value of aragonite saturation. We discussed how depth makes a difference, so we can look at how different depths and different levels of biomineralization occurring at, from surface to depth may vary. We can look at the diffusion of carbon dioxide which may come out of a particular natural feature. We can also experimentally make manipulations, and we can try and get at acclimation, or how an animal during its lifetime can handle the change in its pH. Or we can try and look at adaptation, which is looking over multiple generations of organisms and looking at actual evolutionary change that may be heritable from one generation to the successive generations. So I'm going to focus first on two cases where we look at point source diffusion, and then we're going to look at adaptation and acclimation as well. So our case study one is a really interesting region of Italy. There are places in the world where carbon dioxide naturally bubbles out from under the Earth's crust, and but there are very few places where this occurs in the ocean. So Italy is one of those places. There are places in Mexico and also places in Papua New Guinea. Part of the appeal of doing this research in Italy, even though there are no coral reefs there, is reflects the northern hemisphere and Western scientific legacy bias. The fact is, we know a lot about the environment of the Mediterranean already. We know a lot about the ecology. We know about the natural current conditions there. In some place like Papua New Guinea, where we have coral reefs, there's also a lot less infrastructure. It's a lot harder for most people besides Australians to get to. So there is research going on there right now, but it's still in its infancy relative to a lot of this research that's gone on in Italy. So it's a really interesting spot. It's in the vicinity of this island called Ischia off the coast of Naples. Right here is Mount Vesuvius, and right here is Pompeii. So we know there's a lot of geological activity right around here. And so underwater right here, we have these natural carbon dioxide vents where the carbon dioxide just bubbles out from the Earth's crust into the water. And so when we do the measurements of the pH of the seawater in those areas, in areas further away from the vents where the acidic water is not circulating to, the pH is around 8. And then as you get closer and closer to these carbon dioxide venting areas, you can see that it actually gets quite low. It gets as low as 6.5 in some times of the year, that you can have this fluctuation in the rate of carbon dioxide bubbling out. And that, in turn, causes that seawater pH to fluctuate on these really massive scales. And so people can use the fact that there's a gradient of high pH, normal seawater, just 500 feet away, and then use the vicinity of these vents as a proxy, as a model to look at what future conditions of more acid ocean might be like. And so they go in, they characterize, okay, let's count the types of organisms we see in these different regions. Let's see whether we have the same abundances of worms that don't calcify their bodies compared to snails and clams and scallops that do calcify their bodies near the vents and away from the vents. And right here we have very strong data that shows that in extreme proximity to these carbon dioxide vents, there are almost no gastropods and almost no decapods. So none of these organisms that can be considered highly calcified. Whereas the worms, they seem to not really care so much. They're found all the way across the board. The presence of the bivalves is actually really interesting. There are particular species of mussels that their shells are actually protected by a thin layer of protein, and we think that that actually may help them survive in the vicinity of these vents. We also know that they've had time to adapt to the surrounding region, because these vents have probably been venting for thousands and 10,000s of years and possibly longer. And we can look at the basic ecological measures of the health and strength of an ecosystem by looking at species richness, by how many different species are found away from the vents versus close to them, how many individuals of animals. So it looks like there's the same numbers of individuals close and far from the vents. But if we look at the number of different species and how big the biomass is. So are there lots of large organisms near those vents, or are they lots of teeny, teeny, tiny organisms? Well, the biomass is relatively low close to these vents. So even though there are lots of individuals, there are lots of very small animals. And so we can see where we've got an overall loss of complexity of the ecosystem. We've lost larger animals, we have lost the diversity of different species, but we have a high abundance of very few species, the species that can tolerate that high carbon dioxide. In another set of experiments, they actually went in and they made it experimental plots. Now, this is a fairly complex diagram, but we're looking at differences between the ambient, the high pH, the normal conditions, and then low pH, and then extreme low pH as we get closer to the vents. And they went in and they took a little square of the bottom and they counted. They took a photo and they counted everything that was inside that little square. And then they went into certain other squares and they actually removed everything from that square. So the plot is 20 centimeters by 20 centimeters. So it's not too big, but and they looked at what organisms came back to recolonize that little bare patch of ocean floor. And what they've been able to show is that, in addition to the fact that you have high diversity in these undisturbed plots, once you clear them out, you manage to restore a lot of that diversity. The organisms will naturally recolonize that area. And as you go to lower and lower pH, you see that diversity start to appear. You find that the plots tend to be dominated by things like biofilms, which are produced by different varieties of bacteria, and filamentous, very simple algas. And the types of algae that actually produce calcium carbonate mineral themselves, so calcified filter feeding animals and calcified turf algae, calcareous algae, encrusting algae, all of these varieties of organisms which are present in ambient pH conditions, they tend to disappear in the low and extreme low pH conditions, and they're unable to colonize bare patches of the ocean floor. So moving to a different approach in research, people have looked at coccolithophores. So there are this variety of phytoplankton that is extremely abundant. They photosynthesize themselves, so when they're in the ocean, they're actually making it much more basic around them by sucking up all that carbon dioxide, turning it into sugars, but then also turning it into these little calcium carbonate plates. And they're found everywhere in the surface ocean, but when they become really, really abundant, you can actually see the blooms of these phytoplankton via satellite imagery. They actually change the color of the ocean in those regions. And conveniently, they have also been domesticated for scientific research, so they can be cultured in the lab. And so what people have done is they've conducted experimental evolution. They take lab strains of these organisms and they grow them for many, many generations under different carbon dioxide regimes. And they can show that there are organisms that are able to adapt to these higher carbon dioxide conditions. And there's recent data from long-term time series looking at phytoplankton abundance that show there's actually a trend of increasing numbers of these coccolithophores, and we think that's in part because there's more carbon dioxide. So more carbon dioxide plus sunlight means that they can convert more of that carbon dioxide into sugar. But remember, this is only happening in that thin skin layer of the ocean. So what's good for some organisms is not so good for the other organisms, as we've seen in this example from the events where calcifying animals do poorly under conditions of higher CO2, but we're seeing certain organisms, these types of phytoplankton, doing slightly better under these current conditions. So, one of the things that people are concerned about is the idea of, quote, winners and losers in the process of climate change. I'm going to skip this one. And then I'm going to move to our third case study, so bivalves. Bivalves are particularly important to the Pacific Northwest, but also globally as an important source of seafood, as an important ecosystem engineer. So oysters and mussels are able to generate hard substrate for other organisms to live amongst. They are creating these hard shells that other animals want to live on and among. They can profoundly affect the ecology of these coastal regions worldwide, in addition to being an important food source for human beings and marine organisms. So in the Pacific Northwest in particular and Pacific coast, people are interested in a few particular species. So the oyster that is grown commercially is Chrysostrea gigas. This is an introduced species from Asia. It is introduced in part because when settlers came to the West Coast, they virtually wiped out most of the native oyster reefs. So the native oyster is a completely different species. And once they were wiped out, people were like, wait, we still want to eat oysters. What are we going to do now? So they started importing species of oysters from Asia. And so the common oysters that you will see in oyster bars and in other sorts of oyster food products up and down the Pacific coast, they're going to be this introduced species. There's also different species of mussels that are of interest. So the mussel Middleus californicus is the sea mussel that you see out at the coast. You drive out to the Olympic National Park coastline, and these are the mussels that you'll see in the tide pools. There's another mussel that has been introduced from Europe and is used in cultivation, and it's Mytilus galliprovincialis. So if anyone has ever purchased, mussels from I think one of the local farms is up in Penn Cove. So Penn Cove mussels, all they do is produce mussels. Those are cultured mussels of this species. So you may or may not have heard that hatcheries for these bivalves on the West Coast have been having issues where the early life stages of these bivalves are not surviving the hatchery process, and that is causing a lot of consternation among shellfish farmers about how they're going to be able to sustain their businesses, but also possible consequences for the natural populations of these organisms. And a lot of research that's been done in the past decade has shown that the early development of these bivalves is highly sensitive to changes in carbonate. And initially, people people heard about ocean acidification, and they assumed that since pH itself has so many effects biologically, that they assumed that pH would be sort of like a master controlling variable. And so in this study by Kurihara, she experimentally just added acid to the seawater and grew them up. So this is what a normal mussel larva looks like, and this is what one looks like when it's been grown under these low pH conditions. And here is the a fluorescent dye that's been added to be able to see the calcium carbonate shell, and this is what a healthy one looks like with this intense, vibrant green fluorescence and a very distinctive crystalline pattern called the Maltese cross. And then under these conditions, it's looking not so hot. It's not forming properly into its crystalline shapes. And then you can also see that they're a bit smaller. So those were the very earliest studies that informed us that these early life stages could potentially be very sensitive. And as our interest has increased in the issues surrounding these hatchery failures and the implications for the shellfish industry globally, we've also tried to increase the sophistication of our understanding of how the carbonate system affects these early developmental stages. So over at the Oregon State U, they have actually been trying to manipulate both the aragonite saturation, so the ability to create that calcium carbonate mineral by adding different ratios of bicarbonate and carbonate mineral, as well as managing it by adding in particular concentrations of dissolved CO2 gas. And by doing those manipulations in these different chemical ways, instead of just adding acid and manipulating only pH, they're able to sort of tease apart the different components of the carbonate system that may actually matter to these bivalves. So this is another complicated graph, but they're showing that they've changed the aragonite saturation levels, and they've manipulated the carbon dioxide partial pressure values to try and get a bunch of different treatments that have roughly the same partial pressure of carbon dioxide, but different values of aragonite. So that means that even if the level of dissolved CO2 gas is high, that there are other of these dissolved carbonate components available that are also abundant enough that the ability to form this calcium carbonate mineral is also high. So they're able to chemically manipulate the seawater system through these different independent carbon dioxide carbonate components. And here under the star is the control conditions. So natural Oregon seawater at a pH of around a 0.25 with a aragonite saturation level of around 3.2 and atmospheric levels of carbon dioxide. So all of these gray dots show the types of experimental treatments being done and compared against this control value here. And by doing all these manipulations, they've been actually able to show that the size of these teeny tiny baby mussels and oysters are actually not that determined by just the level of dissolved carbon dioxide gas and not just the level of pH, but actually showing a clear, distinct relationship between the level of aragonite saturation and the amount of shell growth. So this is a really important new piece of knowledge that we're gaining from these studies, that pH may not be a master variable when it comes to creating biomineral, that in fact something else, that this aragonite saturation, the ability to create that biomineral chemically may be more important than just the pH levels where we have low pH here relative to the control, but we still have relatively large shells being deposited. So we're becoming more sophisticated in our understanding of the complexity of the carbonate system, and we're hopeful that this will allow the shellfish industry to be able to cope with the changes of a higher carbonate future. And I'm going to finish up with a historical consideration that this whole concept of ocean carbonate change is not brand new, that some of the most famous respiratory physiologists of the turn of the century actually recognized that the role of carbon dioxide on the oceans and on global climate change may actually have really important impacts. So both Svante Arrhenius and August Krogh, who happened to both be Danish, had noticed, had made comments about the quantity of carbonic acid in the ocean. So that's that reaction that chemical reaction I showed. As it increases the geometric progression, augmentation of temperature, global warming, will increase as well. And August Krogh directly related it to the combustion of coal, that the increase in carbon dioxide due to the combustion of coal would increase the percentage of carbonic acid. And therefore, this is something that we have to take from history, that people appreciated that there was this potential long ago. And so this should be, a wake-up call that we need to pay attention now and think about how that projects into the future. And with that, I will take questions.